Hello everyone, welcome to my YouTube channel. I hope you all are fine. So today's video is about active transport. If you haven't watched my previous video on passive transport, go check it out. So active transport is divided into two types. One is the primary active transport and the other is secondary active transport. So active transport, it occurs against the electrochemical gradient, uphill movement of a substance from low concentration state to high concentration state. Remember, we have talked about it in the previous video. And it requires direct input of metabolic energy in the form of ATP, because it is occurring against the electrochemical gradient. So it requires additional source of energy in the form of ATP, and therefore it is active. And third thing is that it is carrier mediated. So facilitate diffusion and active transport. These both were the carrier mediated transport. And active transport is also carrier mediated, so it exhibits the three phenomena we discussed: the stereospecificity, saturation, and competition. Okay, so there is a little bit of difference between the primary and the secondary active transport. So first we are discussing the primary active transport. So in primary active transport, the energy is directly derived from the breakdown of adenosine triphosphate, ATP, or some other high energy phosphate compound. So examples include the sodium potassium ATPase pump, the calcium ATPase pump. Let's talk about this sodium potassium pump, how it works. Okay, so this sodium potassium pump, it has three receptor sites for the binding of sodium ions on the portion of the protein that protrudes to the inside of the cell, and it has two receptor sites for the potassium ions on the outside. So it has three receptor sites for the binding of sodium on the inside of the cell, whereas two receptor sites for potassium ions on the outside. And the inside portion of the protein has this ATPase activity. And this pump is responsible for maintaining the concentration of sodium and potassium against the concentration gradient. We know that sodium is high in the extracellular environment, whereas potassium is more inside the cell. So when two potassium ions bind on the outside of the carrier protein and three sodium ions bind on the inside, this ATPase activity or this ATPase function of the protein, it becomes activated. Now what happens that this leaves one molecule of ATP, splitting it into adenosine diphosphate and liberating the high energy phosphate bond of energy. So this energy that is liberated is believed to cause a chemical and conformational change in the protein, and thus extruding the three sodium ions outside and two potassium ions to the inside against the concentration gradient. Okay, back to BRS physiology. The sodium potassium ATPase pump in cell membranes transports sodium from intracellular to extracellular fluid, and potassium from extracellular to intracellular fluid. It maintains low intracellular sodium and high intracellular potassium. Both sodium and potassium are transported against their electrochemical gradients, simple, and energy is provided from the terminal phosphate bond of ATP. Okay, next we have the calcium ATPase pump or calcium pump that is in the sarcoplasmic reticulum or cell membranes, and it also transports calcium against the electrochemical gradient. Another example is the hydrogen potassium ATPase pump or proton pump in the gastropyramidal cells and the renal alpha intercalated cells, which transports hydrogen into the lumen of the stomach or renal tubule, and that is also against the electrochemical gradient. So whether it is sodium potassium pump or this hydrogen potassium ATPase pump, the similarity is that they all are working, or they all are pumping the substances against the electrochemical gradient, and they also require this direct source of energy by the breakdown of ATP. Okay, so let's talk about the secondary active transport. In secondary active transport, the energy is derived secondarily from the energy that is originally created by the primary active transport. And it is of two types, counter transport and co-transport. So co-transport is when the solutes move in the same direction across the cell membrane, and counter transport is if the solutes move in the opposite directions across the cell membranes. Okay, so in the primary active transport, we have seen that the sodium ions are transported out of cells, and therefore a large concentration gradient of sodium ions across the membrane usually develops. This means that there is high concentration of sodium outside the cell and low concentration inside. So this sodium gradient, it represents a storehouse of energy, because excess sodium outside the cell membrane is always attempting to diffuse to the interior. That is a downhill movement of a substance from a higher concentration to low concentration. So because sodium is always trying to move inside the cell, so it will carry other substances along with it. Again, if the two substances, if sodium and the other substance is moving in the same direction across the cell membrane, it is co-transport. And if the sodium and the other substance, it is carrying along with it, if they are moving in the opposite direction, then it is counter-transport. Okay, let's see what's written in the BRS physiology. Secondary active transport, the transport of two or more solutes is coupled. One of a solute, usually sodium, it is transported downhill. So sodium is always, it is transported downhill, and it provides the energy for the uphill transport of the other solute. The other substance is moving uphill because it is the active transport. So sodium is always moving downhill, whereas the other substance, it is transported uphill. Metabolic energy is not provided directly by the breakdown of ATP, as we have seen in the primary active transport, but the energy is derived indirectly by the sodium gradient that is maintained across the cell membranes. Another important thing is that if we inhibit the sodium potassium ATPase pump, this will decrease the transport of sodium out of the cell, and this decreases the transmembrane sodium gradient and eventually inhibit the secondary active transport, because secondary active transport depends upon the sodium gradient. So what happens that if we inhibit the sodium potassium pump, the secondary active transport will not occur. Okay, so let's see the counter transport and co-transport in detail. So the example of co-transport is the sodium glucose co-transport. We also have sodium amino acid co-transport. So in case of sodium glucose co-transport, the carrier for sodium glucose co-transport, it is located in the luminal membrane of the intestinal mucosa and renal proximal tubular cells. So what happens that glucose is transported uphill and sodium is transported downhill. Where is the energy coming from? The energy is derived from the downhill movement of sodium, and the inwardly directed sodium gradient, it is maintained by the sodium potassium pump. We know that. And if we poison this sodium potassium pump, if we inhibit this pump, this will decrease the transmembrane sodium gradient, and consequently this will inhibit the sodium glucose co-transport. So both sodium and glucose are moving in the same direction, that is into the cells, into the intestinal cell, into the proximal tubular cell, and the energy is coming from the sodium gradient. So this is co-transport. What about the counter transport? So we have sodium calcium exchange or the sodium hydrogen exchange. So what happens that many cell membranes, they contain a sodium calcium exchanger that transports the calcium uphill from low intracellular calcium to high extracellular calcium. So now calcium is moving outside the cell against the concentration gradient, and sodium is always moving inside the cell because of the sodium gradient that is developed by the sodium potassium pump. So the energy is derived from the downhill movement of sodium. As with the co-transport, the inwardly directed sodium gradient is maintained by the sodium potassium pump, and if we inhibit this pump, there will be no sodium calcium exchange mechanism. So that's simple. Okay, so we have completed the transport across cell membranes. Let's summarize it. Here's a table from BRS physiology. So simple diffusion and facilitated diffusion, they both are downhill, and simple diffusion is the only transport across the membrane that is not carrier mediated, whereas facilitated diffusion and primary and secondary active transport, they require carrier proteins. Metabolic energy, if we talk about metabolic energy, then only in active transport we require metabolic energy in the form of ATP. If it is a primary active transport, then direct breakdown of ATP, whereas in the secondary active transport we have the sodium gradient phenomena that we discussed. And Yes, sodium gradient, it is only required for the core transport and the counter transport, whereas in the primary active transport there is a direct source of energy from ATP. And inhibition of sodium potassium pump, this will affect the three mechanisms: the primary active transport, the core transport, and the counter transport in the secondary active transport mechanism. So that's it for today. In the next video we will talk about osmosis. Do like, share, and subscribe to my channel. See you in the next video.